Welcome back, everyone. I hope you had a good lunch, if a slightly brief lunch break. So we're heading back into our mini symposium on epigenetics and epitranscriptomics, and I will turn it back over to Trevor to introduce our next speaker.
Great. Thank you, David, and welcome back, everyone. As David said, hope you enjoyed your lunch or are still enjoying your lunch. Our first speaker of the second half is Dr. Tom Begley from SUNY Albany. Dr. Begley is a distinguished professor of biological sciences. He's been a long time NIEHS supported investigator, and also I think one of the first ones awards that we had organized here. And he's done a lot of work with respect to tRNAs. And so without further ado, Dr. Begley, please.
Trevor, thanks for the introduction. And I want to thank all of NIEHS for the invite. It's really my honor to be here. Also, a big thank you to Fred Tyson, who's the epi champion and a real leader in helping us move the field forward. I was part of the first class of ones in 2006, so I am feeling a bit old, but I'll consider it experience more than anything. Happy to tell you a little bit about what my lab is doing with regards to tRNA modifications and the environmental stress response. And we've been very much interested in understanding how the epitranscriptome, the collection of RNA modifications, is dynamically regulated in response to stress to regulate translation. And we use computational cell and animal-based systems to, build rules for gene regulation. Just a couple of disclosures. Here's two companies we started to utilize technology associated with stress-induced translational regulation for improved biological manufacturing and also therapeutics to treat disease. I'll fly through this slide as I know the whole group is well aware that environmental stress is linked to human diseases and aging, and this stress can come from both environmental and cellular sources. My lab is particularly interested in how cells respond to reactive oxygen species, because we know if this is left unabated, this can lead to lipid peroxidation, protein oxidation, and DNA damage, which are precursor events that can promote mutations and cell death associated with a number of different diseases like cancer and degenerative conditions. So we're interested in how cells respond to stress, and that's just thinking about the level of at the level of gene expression. We're all familiar with the central dogma, and I just want to highlight, this gives us a blueprint for how genes are coded and turned into mRNA and protein. But from an environmental exposure perspective, we're really interested in when genes are turned on and how much of the gene is actually being made into a working protein. So we know epigenetic signals can regulate transcription. We're primarily interested in epitranscriptomic signals, which are RNA modifications that can regulate translation. And we take a translation-centric view here. We have a couple simple RNA modifications for the U's, the G's, the A's, and the C's shown here, like M5U, M1A, and M5C. But it's important to note there's lots of different modifications, and they're on every RNA species. I think Fred mentioned this at the beginning of his talk, and I'll highlight the RNA modifications on tRNA because each tRNA species it has about 8 to 12 different RNA modifications throughout its structure. tRNA is the most heavily modified nucleic acid. And we primarily like to think about RNA modifications in the anticodon. Position 34 is considered the wobble position, and the anticodon interfaces with mRNA codons during translation. So dynamic changes to RNA modifications at the wobble position have the potential to regulate translation, with the rules for this regulation really being, codon usage. So just a little bit of highlight before we get to codon usage. Studying RNA modifications in tRNA is complicated. Each species has about 8 to 12 RNA modifications. Each organism has at least 50 distinct RNA modifications in their tRNA. So using some of the next generation sequencing approaches has proved fruitful for looking at specific RNA modifications, but studying the collection of modifications requires mass spectrometry at this point. I had the benefit of collaborating with Pete Didon from MIT early in my career. I want to highlight that Pete is an amazing analytical chemist and did a lot of his early work on looking at DNA adducts. And the RNA modification field has greatly benefited from scientists who studied adducts in DNA, like Pete. So essentially what we wanted to do was develop a technology that would allow us to look at RNA modifications in mass. And we wanted to do this in cells that were growing happily or cells that were actually stressed. So essentially what we end up doing is we'll take these two types of cells and we'll individually purify RNA from both of them. The RNA is then hydrolyzed, it's separated based on HPLC, it's identified and quantified by mass spectrometry. What I'm showing you here on the right is a heat map based visualization of about 25 RNA modifications and how they change in response to different types of stresses. Essentially what we end up doing is we get this quantitative data and we compare it to untreated cells. And that's what you're seeing here in the heat map. So what you'll notice here from a simple experiment we've done in budding yeast was that in response to hydrogen peroxide, you get a very specific and a coordinated response of 3 RNA modifications going up, your M5C, your M22G, and your CM. But what I want you to take away from this heat map is that each toxagon has a very specific modification profile that changes. You can have an alkylation specific profile compared to the hydrogen peroxide specific profile. So in trying to understand what these changes at the tRNA level do, we have to think about codons. So the tRNA is the adapter molecule that interfaces really anticodons with codons. And it turns out the anticodons can be very heavily modified at the position 34 and just outside of position 37. And this can influence base pairing. If we think about the genetic code, we've all seen this, probably in biochemistry or genetics when we took it as students. There's 64 different possible codons. 62 of these codons will code for 21 amino acids. In general, we're taught early on that there's an optimal codon for each amino acid, and this is really just the most frequent one used in an organism. But what we're going to be telling you is that codon optimality actually changes as a function of stress. One synonymous codon for tyrosine, say UAU, will be preferred under basal conditions, where UAC, the other synonymous codon, will be preferred under stress conditions. This is very well highlighted for selenecysteine, which is the 21st amino acid, in which a codon is repurposed during stress to improve translation. Now, one of the things about codon usage is that we've been able to show that families of stress response genes possess really unique codon biases. So there's more information in the genetic code than we usually give it credit for. There's regulatory information. This is most, we first showed this back in 2015, when we were working with an enzyme called tRNA methyltransferase 9. This is a writer enzyme that completes the formation of the wobble U modification MCM5U. MCM5U is found on 2 specific tRNAs for arginine or glutamic acid. And what we found is cells deficient in this enzyme are really sensitive to killing by classic DNA damaging agents. They display a cell cycle phenotype. And they really displayed a phenotype as if their DNA damage response was corrupted. We then went on to show that a number of DNA damage response enzymes were overusing AGA and GAA codons, which needed this modification at the WABLU to specifically promote translation. So in the years after that, we started compiling a group of technologies that would allow us to think about translational regulation by the tRNA-based epitranscriptome. I've highlighted the mass spectrometry-based approaches to look at RNA modifications as a coordinated unit. In addition, sequencing technologies were developed to look at all RNAs. One of the things to note is that tRNA is heavily modified. So using RT-based approaches to, sequence traditionally is really difficult. In addition to RNA-seq based approaches, proteomics and analytical approaches need to be used to study the proteome. And in addition, codon analytics based approaches to study the genome and this alternative genetic information at the codon level had to be developed. All of these required a lot of technology that were built in other initiatives. And, it's bringing them all together that really lets us to think about we call codon bias translational regulation. The general model behind this is that in response to stress, you get a change, a writer-induced change in tRNA modification patterns. And what this ends up doing is enhancing the translation of mRNAs with specific codon usage patterns to give you this just-in-time stress response of survival proteins. I will highlight that this codon bias translational regulation it was first established in budding yeast, and our groups and many others in the field now have been able to extend this translational regulation response to stress to other yeasts, infectious disease, parasites, cancer metastasis, and also human cells. I highlight some of the work that we did in mice using naphthalene to show that these writers and RNA modifications are really important for the translational response to this stress. I'll also highlight and just give a little brief update on one of the projects we have ongoing now in the lab. And this is, expanding upon these naphthalene-based results to look at how the epitranscriptome is reprogrammed in mice in response to environmental tobacco smoke. Now, I think we're all aware that ETS has many different toxicants. What we're doing now in the lab is studying how mouse lung and liver respond to ETS exposures. We're doing this in collaboration with Jinjin Ding at the University of Arizona and also Pete Didon at MIT. What I'm showing you down here on the bottom are LD curves for mouse embryonic fibroblasts exposed to different types of ETX toxicants that include naphthalene and formaldehyde, sodium arsenide, NNK, cadmium chloride, and also hydrogen peroxide in there. One of the things that we're doing from these systems and namely the wild type is we're purifying small RNAs and analyzing for changes in the tRNA epitranscriptome in these cell-based models. Now, this heat map that I'm showing you on the right is got lots of information with regards to modifications going up and down in response to each of the individual exposures. What you'll see is these are complex changes in the tRNA epitranscriptome. Now, the big question that we have is, how does this affect gene expression? And what's going to be recapitulated in vivo in the lungs and liver? I will highlight that one of these modifications, Q, which is a very exotic modified wobble G modification, is upregulated in response to formaldehyde. And I'll show you what we think that's doing mechanistically a little bit later. Now, to highlight the complexity of studying translational regulation and the tRNA-based epitranscriptome, just want to highlight that mice has 401 tRNA genes encoding 41 different isoacceptors. Each of these acceptors have 8 to 12 modifications, and they can come from a collection of over 50 possible marks. So it's a big data analytics question and a big data analytics problem. We've been concentrating on developing some simple rules that we can try and apply to these big data analytics. And, at this point, I can transition away from the ETS exposure study and just highlight some of the basic rules that we've been able to figure out so far. But to do this, I'm going to focus on the wobble U modification. The wobble U is chemically diverse in mice and then also humans. These are some of the specific forms. Now, what I want to highlight is that this wobble U is really important for. Sorry about that. My computer is frozen. Oh, there you go. The wobble U is really important for selenocysteine incorporation. Now, Selenocysteine is a 21st amino acid. There's about 25 different selenoproteins found in mice and humans. Now, selenocysteine is incorporated using cis and trans factors found on the mRNA for the selenoproteins, and also on specific tRNAs used to decode this internal stop codon. So selenocysteine will use an internal stop codon, UGA, that pairs with a very special tRNA that's modified to have these wobble U's to promote the incorporation of selenocysteine into selenoproteins. In addition to these regulatory components, there's a CECUS element and the 3' UTR that brings along some very specialized translation factors to promote this. Now, I'll highlight that selenoproteins play really important roles in redox signaling and the removal of reactive oxygen species, and they'll include your glutathione peroxidases and your thyrodoxine reductases. Ooh. Okay, so sorry about that. Now, one of the things that we wanted to actually do is start thinking about some of the codon usage rules associated with selenoproteins. So my lab has been developing analytical approaches for looking at what we call gene-specific codon usage. So these analytical approaches essentially entail counting codons, determining frequencies, and determining statistical over or under usage values in a gene relative to genome averages. We can also take groups of genes and ask, as a group, is this group over or under using specific codons? So this is the exercise that we did for mRNAs for selenoproteins. Now, selenoproteins are a good example of codon bias translational regulation because they're using twice as many stop codons as you'd expect. In this case, it's UGA, and as a group, we use this T statistic for all the 25 mRNAs for selenoproteins. And as a group, they're significantly overusing this UGA. In addition, what you'll notice is they're overusing lots of other codons, primarily that end in what we say is C's and G's. So they're using one codon from a synonymous set more than they should be. On the far right here, we're looking at a heat map that looks at codon frequency for these mRNAs for selenoproteins. And you see very distinct patterning. This is what the big data is telling us, is that a very distinct codon patterns can be elucidated in the mRNAs for selenoproteins. They're typified by this overusage of the stop codon, but there's other codons that are brought along, suggesting there's a very distinct type of translation that's going to be required for these mRNAs. Now, one of the things that we've coupled this codon analytics work to is work with a mouse deficient in one of the writers for the wobble U. This is a ALPBH8 writer. It has a S-adenosine and methyltransferase domain that puts a methyl group onto the wobble U to complete the formation of it. We generated mouse embryonic as expected, the writer deficient mess were quite sensitive to hydrogen peroxide. We also looked at one of the very specific modifications found on tRNA selenocysteine. This is the MCM5UM. What we were able to show is that in response to hydrogen peroxide, the wild type maps see a significant increase in MCM5UM at 20 hours, and there's a significant in an example of a signaling loop where you'd get increased RNA model. And our prediction is that driving the translation proteins. So what you're going to want to look at here is the selenoprotein, looked at the levels of the GPX1 mRNA and wild type and ALPBH deficient mice. Same, actually the ALPBH deficient mice has a little bit more of the corresponding mRNA. On the right-hand side, you see a classic sign of a translational defect. You'll see that the GPX1 protein levels in the deficient maps are significantly down for GPX1 under basal conditions. GPX1 is increase in response to hydrogen peroxide stress in the wild type. You see no corresponding increase in the Reiter deficient system. GPX3 is another good example. So the classic phenotype for translational defect is same amount of mRNA, but a lot less of the corresponding protein because it isn't translated as well. So what we also have been able to do is take this mice and do other types of studies with them. Our latest one is using acetaminophen exposure. So this is really a single a high dose of acetaminophen is administered. 6 hours later, we're then studying protein and RNA-based signals coming from it. In this study, we've given acetaminophen at time point equals 0. 6 hours later, we're looking at the biomarker for liver toxicity, ALT, and we're seeing that it's increasing in the wild type maps, and you're seeing a significant increase in the ALPH deficient maps, suggesting the Reiter is important for the response to this toxicant. We see a trending increase in 8-isoprostanes, suggesting that there's a ROS damage. And then if we look at the protein levels for GPX3, one of the selenoproteins we've shown that's dependent on ALPH8 catalyzed tRNA modifications, you see that the wild type system is able to increase the levels of GPX3 about 5-fold, where this is not matched by the ALPH8 deficient mouse. What I want to highlight next really is what we were looking for in this set of studies is, well, what's happening with regards to tRNA modifications? Okay, what I'm going to show you here is, over 25 different tRNA modifications in wild type and ALPH deficient MEFs in the presence or absence of acetaminophen. So what I'll show you with this heat map is everything's relative to wild type untreated. What you'll notice is that wild type, and this is from livers, wild type livers have a significant increase in the number of RNA modifications in response to acetaminophen. Whereas the ALPH8 deficient livers, you see that essentially it's decreased. We think there's actually sort of a global tRNA degradation phenotype occurring in response to stress. But if you look at some very specific RNA modifications in our system, you can see that MCM5U, which is catalyzed by ALPH8, you see that in the deficient system, the red bars, you'll see that it's flat across the board. But in the wild type livers, the MCM5U modification is increased about 5-fold in response to acetaminophen. It's not the only RNA modification that we looked at. I'll highlight that M1A modifications increase in wild type livers in response to acetaminophen. So do I6A and Y. These are all decreased in our Reiter deficient livers. From here, we're able to sort of build a model of what we think is going on. We think that the defect in RNA modifications is going to be affecting glutathione peroxidase levels, and these are the selenoproteins we've looked at. And you see that some of the glutathione peroxidase levels are down, like GPX1 and GPX3, also thyrodoxine reductase 2, but GPX4 isn't. It turns out that the sensitivity of these mRNAs to a translational defect, is likely guided by other factors also, namely total mRNA levels. Here's the basic model that we have is that, ALPH8, WOBLE-U, and other tRNA modifications are playing important roles in adapting to environmental stress. Highlight that, we've seen this with acetaminophen. We've got a published study showing that naphthalene from mothballs is also doing this. And our preliminary data in our ETS project is showing this is the case too. So what we think is going on is these Reiters and modifications in our tRNA are helping to promote the translation of proteins that are really important for acute and chronic stresses. Now, I want to say it's more than just WOBLE-U and ALPH8 that's playing a role. There's lots of different RNA modifications. And I'm going to transition here to another story, namely in our HepG2 human hepatocellular carcinoma cells, because it highlights that the WOBLE-G modification, which is this cuisine moiety that I talked about earlier, is also really important for responses to stress. This is a study in which we've treated Hep G2 cells with sodium arsenite. Sodium arsenite is what we're able to show in our human systems is that the modification Q is dramatically increased in response to sodium arsenite. Okay. In a LD20 dose, this happens after 6 hours. If you increase the dose, you can actually see it as little as 1 hour. Now Q is a really interesting RNA modification. It's found on tRNAs for tyrosine, histidine, asparagine, and aspartic acid. I'll highlight that each of these corresponding tRNAs can decode a U or C ending codon for the corresponding amino acid. Now, you know, the interesting thing about Q in tRNA is that humans and all mammals require a little help to get it into their tRNA. So Q in tRNA is inserted by the QTRT enzyme. This is a transglycosylase. And essentially what it's going to do is it's going to use a Q base shown here, and it's going to swap it in with G to make Qine in the tRNA. So it turns out mammals don't make the Q base. We get it from our diet and also from our gut bacteria. Gut bacteria will take guanosine and make this Q base, and then we use it. So it's really a great example of an epitranscriptomic mark that's linked to diet and the microbiome, which I think is going to be a big area of research in the future. So you were using cell culture-based systems to study Q-acine and tRNA, and it turns out that the Q base is actually found in your FBS. So what we can actually do is we can dialyze FBS and remove the Q base and grow our cells in this dialyzed FBS. We can also buy the Q base from Sigma, and we can put it back in the FBS to replenish the system. So here's an experiment. We have our cells grown in normal growth medium, which we call as sort of our normal Q. We have them in dialyzed FBS growth medium, which is the low Q. And then we have them in our dialyzed FBS plus Q, which is our greater than normal Q. We've gone in and we've purified the tRNA from the cells grown under these conditions. And you can see about 100-fold decrease in Q in the dialyzed FBS, and it goes up above, sort of normal levels when we add the Q back in the system. If we start looking at the corresponding cells, you can actually see that the cells that are grown in the dialyzed FBS have a really decreased viability in response to sodium arsenite. And then this is rescued as soon as you add the Q back into the system, shown in the blue. We go further to actually study the effects of, sort of low Q in tRNA. What I'm going to do is just highlight some data that we have measuring reactive oxygen species levels and mitochondrial mass in these systems. What you notice is that the cells grown in this low Q condition in the tRNA, you see actually a significant increase in ROS via cell rocks orange in response to sodium arsenite that's rescued as soon as the Q gets back into the tRNA. In addition, we've looked at mitochondrial mass, and you see that, the cells grown in this dialyzed FBS with low Q in the tRNA have a decreased mitotracker green signal, which, correlates to decreased mitochondrial mass. And this is further exacerbated in the presence of sodium arsenite. So next thing we want to actually do is look, well, what's going on at the level of translation? What I'm showing you here are polysome profiles, which is a way to measure actively translated mRNAs by the number of essentially ribosomes that are bound to them. This is a really busy figure, but what I want you to take notice of is the polysome profile in red. This corresponds to the cells that are grown into dialyzed FBS and treated with sodium arsenite. What you can see is that the levels of actively translated mRNAs dramatically decreases. On the right-hand side, what I'm showing you is just a normalized polysome plot where we take the cells grown in a treated condition and normalize it to the untreated condition. And what you see is these, the cells grown in the low Q condition have a significant decrease in the number of bound ribosomes that are translating mRNAs. So removing Q from tRNA actually has this massive effect on translation. And If we go and start thinking about the model that we're putting forth on it, the general idea is that it has such a big effect on translations because so many of the corresponding codons that need to be translated are going to be disrupted. Okay. In general, we think that translation is perturbed in a very specific way. Using some computational and wet bench studies, we've been able to, correlate this decrease in translation to transcripts that, overuse the C ending codons for tyrosine, histidine, asparagine, and aspartic acid. So this is a model in which Q is promoting the decoding of the C ending codons in specific mRNAs. Now, this is a sort of a complicated model, and what I sort of want to highlight is is going forward to study translation, it's complicated. So what we really need to be able to do is integrate lots of different data types and think about translation from what we call as a gene-specific codon metrics perspective. So we've been actually downloading and looking at all the genes in an organism to look at gene-specific codon patterns, developing quantitative data related to these codon patterns, and sorting these patterns into into different groups. We use clustering-based approaches, heat maps. We've also used some machine learning approaches. But what I'm going to highlight here is this is just an example organism that has 20,000 plus genes. We've analyzed gene-specific codon usage patterns to get quantitative metrics on each of the codon usage in a gene. And then we've built these heat maps, which show you patterning. Okay, and the general model that we have is that each one of these patterns is like a QR code for translation. And to match this QR code, you're going to have specific sets of translational decoders that are found in collections of RNA modifications. So this blue QR code, essentially would be, translated by a signature of modifications rich in wobble U's and I6A, where in corresponding this red QR code, which corresponds to this group here, would be decoded by RNA modifications efficiently that have a lot of this wobble G, the Q and a Y, and so on. So, this is how we're thinking about translational regulation and stress responses. I want to thank my group and all my collaborations for helping with these projects and working with me. And I'm going to highlight Pete Didon, who's been my long-term collaborator from MIT. Xinjin Ding from the University of Arizona has played a really important role in our exposure studies. I want to highlight my current group now and all the past members. And I want to give a big thank you to NAEHS for all the support. I couldn't have done it without them, and I really enjoy working with them. So thank everybody, and stop there.
Thank you very much. That's terrific. And the world keeps getting more complicated. Absolutely. So we have perhaps time for a single question, if there's one now, or we can wait until we have the general discussion. This we're back on time, on our modified time. Let's see. Okay. So maybe I'll take this opportunity rather. So with the interaction on that sort of the very massive impact that you saw on translation in the Q example, is there feedback in terms of the regulation of the modification then?
So yeah, I think there's going to be some feedback. So there's definitely transcriptional feedback. So I think that one of the things that we're seeing in the field is that a lot of transcription factors are going to be regulated by what we call as this codon bias translational regulation, so that you can sort of match transcriptional programs to changes in translation. So you'll see the loops like that. There's going to be other subtler forms of feedback too. We're starting to see some translational feedback also that comes into play, but I think the transcriptional feedback is going to be the much bigger signal that's built into the system.
Great. Well, for those of us that work in the nucleus, that's always good to hear. Thank you very much.
Sure.
Yeah. So we'll move on to the next presentation, which comes from Dr. Yunsum Nam at UT Southwestern, where she is an associate professor and is internationally recognized structural biologist, particularly focusing on modifications around RNA structure. And her talk today will be about looking at understanding some of the mechanisms around tRNA methylation. Dr. Nam, thank you.
Thank you for the kind introduction, and thank you for having me here today. I'm a little bit under the weather, so please tell me if you cannot hear me very well. Yes, my name is Yun-Sun Nam. I'm at University of Texas Southwestern Medical Center. I am a biochemist and a structural biologist, and we study how RNAs and proteins communicate with each other. So the title of the talk today is on substrate recognition by modification enzymes. Can you see my slide? Okay.
Yes, we can.
Okay, great. So as we have discussed today, RNA function, RNA is important, and it can do so many things, and we see its role is important for in responding to the environment. But RNA function can depend on many factors, like the RNA sequence and the structure that it can take, both of which can affect RNA localization and interactions with other molecules. And all of this together would result in some sort of a biological outcome, right? So my lab is interested in how RNAs and proteins interact with each other, because that is involved in all of these processes. And as we are discussing today, RNA modification is a very important part of RNA entity. So it gives it a big portion of its identity that would affect the biological outcome. So my talk is going to, today's talk is going to focus on how the RNA sequence and RNA structure affect the modification process. Okay, so just a very brief background. RNA helices look very different from DNA helices. DNAs have these grooves where you can read the sequence. For RNA helices, you cannot access those grooves. So sequence is really difficult to read, but RNAs nevertheless have a lot of structures, including helices, and therefore proteins have to recognize RNA structures. RNA structures are very dynamic, which makes it difficult to really pin down what's important. And then when you actually figure out the complex, they can actually look different again in the complex context. And what's causing the RNA modification event is this active complex. So this is what we focus on. How, where is the specificity coming from? Where is the modification efficiency coming from? And how do you have enough affinity? And how is this whole thing regulated? So the first story I'm going to talk about is META1 WDR4 effect on tRNAs. So the previous talk did a wonderful job. Really, it was a fantastic talk to get you to appreciate how tRNA modification is important in biology. And so the one example I'm going to talk about is modification of 46, which is a different area than the anticodon loop, but this is nevertheless important. So META1 and WDR4 are proteins, and they together modify tRNAs to create M7G modification. And this leads to tRNA stabilization. Now, we started working on this partly because both of these proteins have been associated with human health. So META1 amplification is seen in various types of cancers at pretty high percentages in certain types of cancers. And WDR4, its partner protein, its mutations have been found in kids with brain malformation disease. So what's interesting is both too much or too little methylation are bad. They cause diseases. So how is this protein complex regulated in a very regulated tightly? So we started working on this, trying to understand the mechanisms. And this one, we worked on several different structures of the complex. So these are cryo-EM structures of the protein RNA complex and in different states. And these are crystal structures of the protein complex as well as the catalytic domain with cofactor. And combining all this, what we found out is that there's initially when the molecules come together, this is kind of like a rigid docking. And now when I say rigid docking, it's kind of like these are preformed puzzle pieces that just come together and they just fit right in. And that's because individual crystal structures of the protein subunits or the tRNA, they all look very similar to when you have the full complex. This is pretty unusual, but this might be the reason why the arginine 170 mutation that's in those kids with the brain developmental defect, that's right in the middle of this interface. And if you even have a slight change in the angles of this interface, might have a detrimental effect. Enzymatic function-wise, it's a pretty weak loss of function, but it is, it's about 40 to 50% activity. Okay, so this is what happens initially. Well, how is the tRNA recognized? Most of it is through the elbow region. The tRNAs have this sort of unique elbow region where the D loop and the T loop come together. And this is what we call usually the elbow region distal to the anticodon loop. And this area is recognized through shape recognition and charge complementarity. And you can kind of see the protein cupping that area, cupping that elbow on the front side and the back side. Okay, so we got this complex and we think we know how they come together, but this is not the end of the story. Because look at G46, this is the base that needs to get modified on this tRNA. And it's kind of tucked inside the tRNA core, and it's not available for chemistry. So how is this going to happen? Well, through multiple structures, we were actually able to show that it can unravel, and then this is how you can activate the tRNA for modification. So most tRNAs that we we tested, all of them actually bind the protein with similar affinity. So the tRNA structure is like a basal requirement for the proteins to interact with the tRNA. But there's a higher level requirement that G46 can flip out in this molecule. And that is the next requirement, so only the yellow ones would actually get modified. So what's actually going on here? Well, this G46, like I showed you in the movie, it has to flip out. In this inactive position, G46 is kind of sandwiched between these bases, and it's kind of it's really occluded. It's kind of like hidden in here. And then when it flips out, in order to pay for that cost, the protein has to come in. These arginines actually replace where the G used to be, and then kind of replacing the ring stacking interactions. That's important. So, so the protein is activating the tRNA unfolding to activate the methylation. Okay, how did this protein come out of nowhere, right? So this is the cryo-EM structure of the apo state, and then this is the one with the cofactor. And what happens is that when you have the cofactor, this N-terminal region of the protein gets rigidified. So in the cryo-EM map, we can actually see this whole dark density just pop up. So this is activated once you have the cofactor bound. And that's because the cofactor provides part of the binding surface for this peptide. And hopefully in this view, you can see that this switch peptide, when it becomes rigidified, it becomes the glue between the protein and the RNA. So there's and once it does that, it pulls in this helix from WDR for the partner protein, and then these two can provide a binding scaffold for the RNA. So I went through that pretty fast. So let me show you another movie just to kind of give you that perspective. So these, So these proteins and the tRNA, they come together initially, and then there's binding, recognizing each other's shape. Initially, there's not a lot that's going on. It's except for this loop. But then when you have cofactor, there is this unfolding of the tRNA, and this G can basically dock into the catalytic site. How did that happen? Because once you have the cofactor, this dark peptide becomes ordered on top of the cofactor. And when that binds the cofactor, then it pulls in this helix, and these two together, they interact with each other. And then they hold on to the backside of the RNA. I hope you can see right here. And then that allows the front part of the tRNA to unravel without losing contact with the enzyme. And of course, part of that peptide is what's kind of coming back and paying the cost of flipping out this G. Okay, so from all of this, we learned that there's a lot of structural recognition. There's also a little bit of sequence recognition. The loop that gets modified, it starts to interact with the protein a lot more. So we think that there is some sort of sequence dependence. But as you can see, the sequence of the top one, so AGGUU in this, it'll be reading from right to left. This sequence, it also appears in the bottom, the one that's least methylated in this group. So what I'm saying is the sequence does not necessarily determine the methylation efficiency. And then glutamine, which never gets modified, we actually have put in sort of some of the optimal sequences to it. So this is a wild type glutamine. And then we put all these different sequences to it. And there's very little activity compared to what we saw the structure of, which was lysine tRNA. So the sequence is important. When we mutated, there's a little bit of loss of methylation, but it's really only partially contributing to the methylation efficiency. And a lot of the methylation is dependent on tRNA folding. So I'm gonna show you one more thing. You know, we just heard a great talk on how tRNA modification can be regulated by stress and environmental exposure and things like that, right? So how do these enzymes change their modification efficiency? That's a question. So this peptide that I've been talking about, the switch peptide, it has a serine, and S27 is over here, right next to this arginine. And this is kind of a close-up of this serine. And the serine is modified by a kinase, AKT, which can phosphorylate this serine. And look at how many phosphates there are near that serine. So, of course, it's not surprising that this would be this would make the enzyme less active. So this is not a preferable conformation. So this can the AKT can inactivate metal 1 activity. You know, tRNAs that get modified, the ones with the yellow hat, those are modified. But if you have this phosphomimic, then there's a decrease in methylation activity. Now, there's a surprise here. Not every tRNA behaves this way. There's yet another group of tRNAs. And these tRNAs, there's pretty similar amount of methylation, whether you have this phosphorylation or not. So now if you look at the RNA pool, the tRNA pool, we just heard about how tRNA modification and changes in tRNA stability can change translation. So what this means is when you have AKT activated, this signaling cascade somehow can now change tRNA abundances. So at that point, suddenly these tRNAs would become a lot more abundant, and then these tRNAs would be less abundant. And you're changing the tRNA profiles, which means you're changing codon bias during translation. So I hope I'm going into atomic details, but by going into these atomic details, what I have shown you is how small change in this modification machinery can basically can really change how the RNA modification profile, and that can affect biology. So I hope you got from my talk that a lot of tRNA modification is dependent on structure recognition. So RNA recognition can depend on sequence or structure recognition. And there's kind of a spectrum, whether how much you depend on the sequence recognition and how much you depend on the structure recognition. And the example I showed you would be kind of a mix where tRNA structure really drives a lot of the recognition and enzymatic efficiency. Now, the second part of the talk I'm going to talk about is going to be very focused on structure recognition. But remember, double-stranded RNA, you cannot recognize a sequence. So for the protein to recognize RNA sequence, you need these opportunistic moments when you have single-stranded RNA, whether in a loop or mostly these loops that are connecting secondary structures. So in some ways, to be able to recognize this particular sequence, you require this structural context. Okay. So we've already talked a little bit about, quite a bit about M6A. M6A is probably the most abundant modification for messenger RNAs. It is known to affect RNA stability, translation, splicing, and maybe even chromatin accessibility. And metal 314 is the major modification enzyme that that is responsible for most of the M6A modifications in messenger RNAs. Now, metal 16 is another protein that can add M6A to mRNAs, but it works on pretty specialized targets. And I have, my lab has been studying metal 314, trying to understand how it does this. And back in 2016, we showed that using crystal structures and biochemistry, that metal 3 is the active component and metal 14 is the inactive component. Now, we've also done studies on metal 16, which does the same chemistry, but it looks very different. But from this, we were able to kind of learn from this example and could postulate that this RNA binding should happen kind of around here. In the middle of this putative RNA binding interface, there is arginine 298, which is the most commonly mutated, most frequently mutated residue in cancer patients in metal 14 gene. So there are more mutations on this residue than other areas in metal 14, and the mutations can be 3 different types, and that have been found in different patients. All of these are seem to be heterozygous background, so you don't need, this is a dominant mutation. So we have tested whether this mutation affects cell growth phenotypes. And in vitro cell culture systems, so these are HepG2 cells, liver cancer cells, and you can see that there's increased cell migration with this mutation and not D312A. D312A is a loss of function mutation that we know is right next to arginine 298 in space, and but we always see less activity. So what I'm saying is we see kind of a different phenotype for this mutation compared to this other loss of function mutation. So we thought this was significant. And these cell lines are also, they're expressing about 50% wild type and 50% mutant proteins. We've also tested how this mutation affects tumorigenesis, and this is in collaboration with Hao Zhu's lab with help from Brett, who helped with pathology analysis. So you can see there are bigger tumors and more tumors in mice that got arginine 298 mutation than the wild type or the other allele. So and the arginine 298 causes worse, more progressed tumors with some necrotic tissue. So we were convinced that this mutation is linked to oncogenesis, and we wanted to find out how this actually happens. So how does a cancer mutation affect RNA sequence preference in RNA modification, right? So we had to step back and kind of broaden our strategy, because what if it can affect RNA binding in many different ways, right? So we randomized a library of a segment in the library, and then we incubated them with purified methyltransferase wild type or the mutant. And then we looked for what are the RNAs that bind to the wild type versus the mutant. And so this is basically over 3 rounds of selection, and then we pulled down the RNAs that were selected by the wild type or the mutant proteins. And we found out that all the sequences that were selected by the protein were pretty random. There was no enrichment and not a clear consensus sequence. So we changed our strategy, and we looked at the RNAs that were methylated. So after methylating them, we then pulled down that methylated RNAs and then sequenced those. So from this, we found out that the wild type indeed prefers GGAC sequence, which is very similar to the DRACH consensus sequence that have been found in a lot of epitranscriptome studies. The mutant, the oncogenic mutant, showed a clear enrichment for GGAU sequence, which is different from GGAC. So there are, I think it's very rare to see an RNA enzyme, a protein, to have gained a new specificity with one mutation. So we were interested in how this would happen with the other mutations, and then indeed it has the same change in specificity. The loss of function mutant still maintains the C after A with even the little methylation activity it has. So this is looking at sequencing data, and then we've also looked at enzymatic assays to be quantitative. And then we found out, so these are in vitro methylation assays where we're looking at the amount of methylation. So GGAC something is always methylated the most, whereas these are less modified, okay? So we're just counting the amount of radioactivity incorporated into these RNAs. Well, so the loss of function mutant has just lower activity, but the pattern is very similar. So this is indeed a loss of function mutant. Well, if you look at the arginine 298P mutant, there is higher activity with this GGAU something sequence than the canonical GGAC substrate. And all the arginine 298 mutations that can be possible with one nucleotide change, we were able to see similar change in activities. Therefore, these are gain of function mutations that now have a new biochemical activity where it prefers a new sequence. And we have shown that this doesn't really change in the context of different sequences or different structures. So for the wild type protein, it always prefers the solid bar, which is the GGAC something. And in the mutant, you always prefer it always prefers to methylate the open bar, which contains the GGAU sequence. There are some differences. So it might depend on the structural context, but overall, this sequence preference is maintained. So how is it possible that you have a gain of function with several different mutations of the same residue? And we went back to structural biology and we saw this arginine 298, this residue that gives you a gain of function activity is quite far away from the active site, about 20 angstroms. So we saw the structure of the mutant, and this is the arginine to proline mutation. So when you take away this arginine because of the mutation, then this other arginine in metal 3 comes over to fill in this gap. And then, sorry. And then this histidine also moves in to interact with this arginine. So this area is rearranged. So what can we make sense of this, right? So far, there's no structure of metal 314, a key methyltransferase in complex with RNA. But with all of this biochemical data and structural data, we can propose how an RNA interacts with this enzyme. So this methyl group on the SAM, it's gonna go to this nitrogen on adenine. And then the next nucleotide, C or U, is being detected by this pocket around here. And because of rearrangements in this area, you can start to distinguish between C versus U. Okay, so We have done all of this in test tubes, and you might wonder, is this actually going on in cell culture? We see new peaks, new methylation peaks in the arginine 298P cells. And if you look at the top 5 sites, that's the top 5% of the sites, we indeed see start to see GGAU coming up. This change in RNA methylation triggers a change in gene expression profiles. One of the things that's pretty interesting is that there's the same change. If you do all two-way comparisons, Wnt signaling is definitely the one that's uniquely different in the arginine 298P cells. So I've been talking a lot about this mutant and this mutation. Sure, you can have a somatic mutation and that can be oncogenic. That's interesting, but that's not a very high percentage of patients. So, and can we learn a little bit more from this than that? Well, there's even more insight. Well, you know, the wild type protein prefers C, and then the mutant protein prefers U. But both proteins have some residual activity towards the other. So this is what I call star activity, kind of like the restriction enzymes. So they still have some activity because the control, the negative, the background is so low, you can't even see it here. So, and this means there is some significant amount of activity on this less preferred sequence. So let's go back and look at the model for how oncogenesis can depend on dysregulated methylation. In normal conditions, you have this methyltransferase expressed at normal levels, and it prefers this canonical sequence, and there's methyl groups added to these sites, and fewer methyl groups on the GGAU sites. This is the star activity. There is some, but obviously this is preferred. Now, with this mutant, we found out that it's actually flipped. There are more methylation events on the non-canonical sites and less on the normal sites. Well, you can imagine both of these can be bad, right? What is interesting is a lot of cancers have reported overexpression of wild type. So if you have too much of wild type enzyme, you will get hypermethylation in both of these sites. And what's really interesting is that these bottom two look very more similar than these. So what I'm saying is it's worth looking at these new sites that we discovered with the mutant. And this might have a role in even oncogenic pathway via the overexpressed methyltransferase. Just to remind you, a lot of the previous studies on these different models, different cancer models, have focused on GGAC sites. A lot of the library preparation or bioinformatics workflows actually become blind to the GGAU sites. So this might be worth going back and investigating. So today I have talked about how this sort of dynamic crosstalk between RNAs and proteins are very important in providing the specificity that is important for to control where the modifications are placed. So we're not just talking about more methylation or less methylation. It's about which molecules are actually getting that modification, and maybe even a different position on the same molecule. So with the tRNA, I've talked about how different tRNAs are regulated by because of this dynamic movement, which can change depending on the tRNA sequence. And then in the messenger RNA modification example, there's all of these RNAs, again, bind the same, but it's the chemistry that changes with these changes in the side chains. So this, the tRNA modification changed with the post-translational modification. One phosphate group can trigger the change in which tRNAs are more modified. And then this can arise from a somatic mutation. And we don't know yet what other post-translational modification could also impact this kind of RNA substrate specificity. Both of these systems end up affecting gene regulation in a major way, translation and codon bias, or mRNA stability, and again, translation. So I want to end by saying how differential RNA modification can correlate with disease, and that RNA modification can change dramatically with very small changes in the RNA modification machinery. So you might wonder, like what, we go through all this trouble trying to get the atomic level structures, but it really provides us new insights on how all of this machinery works, so that with some kind of gene mutations or signaling pathways, how these can be regulated and maybe fixed. With that, I'd like to thank all the people who did the work. The first story was led by Victor, and the second story was led by Chi. And we are thankful to our collaborators. I already mentioned their contribution to the mouse work, and all the funding agencies that helped us and supported us through the years. And thank you for your attention.
Dr. Nam, thank you very much. That was really quite elegant talk. We have time for a few questions. Dr. Begley.
So I was just clapping on how impressed I was with it. Really beautiful work. So I don't really have a question, but I was, it really highlights the complexity of what's going on, and it's almost scary how much we have to do. But it was beautiful. Thank you.
I agree.
Great. Thank you, Tom. All right.
Yeah, since I just one last word, wouldn't it be great? Wouldn't it be great if someday we can actually, if we could know some of these rules on what determines whether you're going to get modification, and how each where are the pressure points for each modification event, so that you can, so that we can have an idea when you have a novel RNA, let's say, oh, this RNA is involved in this stress response and it's changing, and then we can just run it through a server and see like, oh, it's going to be modified by this enzyme and that enzyme and this modification, and it might be sensitive to this kind of regulation, so that would be our dream.
I share that dream. That was a great idea.
Dr. Hollian.
I can't hear you.
Andre, you're
Andre, you're muted.
Am I hear me now?
Yes.
You can, yep.
Okay, I had my settings wrong. It was so more of a comment, but I think it really goes to all the structure activity relationships that you mentioned. It drives the interactions between, in this case, as you described, to regulate what is going on, but also goes to many other aspects when it comes down to biology, whether it is particles interacting with membranes, what I look at, or what you're looking at here. That's what defines it. I mean, it's very logical, and to be able to come up with that information is, and how that happens and what the consequences are really critical. Now, I want to ask you the hard question: how are you going to use this to develop therapeutics?
Oh, so one thing is we have shown that if you have this mutant, it's more oncogenic, right? So as you can see in our model, if the mutant can differentiate between C versus U, we think maybe the mutant protein can distinguish, can distinguish between small molecules. So we actually have a small molecule screen so that we can identify inhibitors of this enzyme that preferentially works on the mutant, the oncogenic mutant versus the wild type. This is just one type of thing that we can think of for developing therapeutics. But I guess I'll switch it around for you. Like if you don't know how all the parts work in a car, it's hard to fix a car, right? So, and we first need to kind of understand how all of this works, and then maybe we can find exactly how to control something that's broken, the broken part. So that's kind of that's, I guess, my sort of open-ended attitude about this.
Thank you. Dr. Wang, we have a last question.
Yeah, great talk. Yeah.
Thank
So,
you.
yeah, for the last section, you draw the analogy between say wild-type META3 META14 complex and the mutant META14. You said the wild-type protein may also induce the U containing sequence context, right? Is it possible that other subunits of the META3 complex can also provide more specificity, so that we prefer, say, the AC, GAC, or over GAU, or you think that's not likely?
Yes, that's a great question. Metal 314 can be found in the context of a much bigger complex in the cell, and they are important for substrate specificity in a different way. So when we did all of these experiments, they were in, you can see the enrichment of GGAC pop out with nothing else provided, which means metal 314 is enough to specify that that's the preferred sequence. We think the other components are probably important to localize the methylation events to, like, let's say, near the stop codon and the 3' UTR, because of like their interaction with other machineries in the cell, like the spliceosome and stuff like that. So it's not like every GGAC is modified in the transcriptome, right? So that kind of specificity is kind of dictated by those other factors that interact with metal 314. But locally, the detection of the local sequence, we think is metal 314 is enough, because it's able to come up with this sequence in this randomized RNA search that otherwise it wouldn't be able to do that. Yeah.
Yeah, great. Thank you. Yeah.
Thank you for the question.
Thank you very much, Dr. Nam. I think in this time, we will move on to the final presentation in this symposium. It comes from Dr. Ting Wang at WashU, who's the Stanford and Karen Lowenthal Distinguished Professor of Medicine. And he's also the chair of the genetics department at WashU, and a well-known and important contributor to a number of NIEHS and NIGHRI consortia and groups, including the Target 2 consortium, where he heads the data coordinating center. And Dr. Wang will tell us about the integration of multi-omic data sets. Ting, off to you.
Hello, everyone. Can you hear me okay?
You're coming loud and clear.
And slides are good.
Slides are great.
Awesome. Thank you. Thank you so much for the opportunity to share with you here. And I want to begin by thanking Fred for putting together such a wonderful session. I feel very, very honored to be here and then to share with you some of our own research. There's a lot more I want to thank about Fred, so we'll talk about that a little bit later. So I'm from Washington University in St. Louis. I hope many of you have visited St. Louis. It looks really good on the map, right in the middle of the US map, right? So we're not Kansas City, but St. Louis and Kansas City are both in the state of Missouri. So we consider the Chiefs our home team, right? Go Chiefs, right? What St. Louis does have is this beautiful arch, right? So this is a symbol for the Lewis and Clark Western exploration. We use this symbol for one of the project in the lab that is called WashU Epigenome Browser. Dr. Tyson mentioned that at the very beginning briefly. So we actually use this to present a lot of the epigenomics data that the community has collected. So I'm tasked to discuss the integration of multi-omics data. I think that's because I've been really fortunate to have been involved in several NIH. Of course, NIH has funded large multi-omics data production projects. So my plan is to briefly describe my own personal experience with these consortia projects that are generating multi-omics data for the large scientific community. And I also describe just one case study of my own research that is taking advantage of the publicly available multi-omics data to investigate the so-called junk DNA or the, in our genome. So before we talk about multi-omics data, or even before we talk about genomics, I feel that I should mention the Human Genome Project. Right? So I am a genome scientist. This is the human reference genome when we print them into book volumes. A lot of what we do is to read and understand this book, right? We call this the book of our lives. The ultimate goal of integrative genomics is to understand the meaning of this book. Indeed, I think it is the Human Genome Project that propelled the genomic technology development so that we can do integrated genomics work. So this reference human genome has revolutionized biomedical sciences, and it's clearly very, very impactful. So perhaps it's worthwhile to quickly go back to this, to our memory lane, right? So this book represents the product of the Human Genome Project. It certainly impacted the modern science history quite dramatically. It fundamentally changed how biomedical sciences are being conducted. This is also one of the first big data projects to set precedents for team science, genomics, and data science, right? So this draft genome has been completed multiple times, each time being a major improvement in terms of quality and coverage of the human genome assembly. And of course, many different species got their own genome assemblies as well. So with the incredible investment of short-read data, medical records, and functional studies, this reference genome is shared across genomic community, right? So it is critical for us to make new and meaningful discoveries. Over the past 20 years, legends were born and a story were told about the Human Genome Project. If you count 2003 as the benchmark for the completion of the Human Genome Project, then the last year is the 20-year anniversary. So this so-called complete genome, over the past 20 years, the genome assembly actually continued to be improved and polished by the Genome Reference Consortium. Many probably still remember HG 15, HG 17, 18, and 19, all the way to HG 38 that we all use today if you're doing human genomics studies. But even HG 38 is not complete, right? So it misses about 8% of the sequences. This final gap was recently closed by the telomere to telomere consortium. So I will mention towards the end of my presentation, a new project will once again revolutionize genomics. That is the Human Pan-Genome Project. So what kind of value has the Human Genome Project provided? Today is actually a pretty special day. 11 years ago, on this day, right, President Barack Obama, in his State of the Union address, said, every dollar we invested to map the human genome already returned $150 to our economy, right? So that was the 10th year anniversary of the Human Genome Project. Now another 11 years have passed. The value clearly has increased dramatically. So this is kind of the one map that is very relevant to integrative genomics. Another very relevant map that really got the game started is the human reference epigenome map that at the very beginning, Dr. Fred Tyson also mentioned. So the Roadmap Consortium, as well as the ENCODE, 40-nucleome, IHAC, International Human Epigenome Consortium, and our very own Target Consortium have produced tens of thousands of epigenome data sets, giving us an opportunity to understand the tissue, cell type, developmental stage, as well as environmental specific epigenetic dynamics of cells, right? So my lab actually helped present this data to the public using our WashU epigenome browser. So this is the kind of data that we present. And we also use this data to probe our own interesting questions, including what I'm going to mention in a couple of minutes, the dark region of the genome transposable element. So if we take a step back and then look at how genomics has been evolving, right? All sorts of omics has been evolving together, right? So omics is defined as this method to measure and functionally characterize different biomolecules in cells or tissues. So the past 20 years, we have seen an exciting and rapid evolution of omics technologies, going from genomics to transcriptomics to epitranscriptomics to proteomics, metabolomics, metagenomics, and all the way to exposes as a yet another omics measurement, right? Sequencing-based technology seems to be dominant, even though the field really started with array-based platforms. There are also revolutions in imaging and mass spec and methods for collecting specimens and accompanying, all this evolution of omics technologies. So these new technologies also produce new types of data, right? That require very different skills and paradigms to integrate, to analyze, and to make sense of. We have also seen many waves of computational algorithms and tools development, all the way to the current hype on artificial intelligence, right? Ultimately, the knowledge we gained through integrative analysis will allow us to develop predictable models and development in genomic and epigenomic engineering technologies will make it possible for us to manipulate the genome and epigenome to test those models. So all of these critically impact how we think about defining, identifying, and integrating this vast genomic resources. So I'm gonna now jump into this one case study here, right? So I hope to illustrate how we have integrated publicly available multi-omics data to investigate a part of the genome that most people actually, ignore. This has been long called junk DNA or dark matter of the genome, and that's the part of the genome that is derived from a transposable element. In fact, if you look at the material world, right, so there are dark matters and dark energies, and in our genome, there's also very similar regions of the genome that we call this dark matters, right? So one of the first surprises from the Human Genome Project that we just briefly mentioned was that coding sequences only make up about 1.5% of the genome, whereas close to 50% of the genome are derived from transposable elements, such as signs, lines, and LTRs. If we talk about epigenome, or just the CPG sites, right, more than half of the CPG sites in our genome are derived from transposable elements. These regions are repetitive, and they have long been understudied. You probably read these three statements in thousands of papers, right? So back in the days when we used microarray, the probes often only represents non-repetitive regions, so we did not even have data to begin with. Second generation or short read sequencing start to take over the field, but we only kept reads that are uniquely mapped to the genome, and the rest are discarded, right? So there are a lot of computational work. In fact, my own PhD work was focused on designing multi-finding algorithms. So back then, I focused on the parts of the genome that are repeat masked, right? So that's the first thing we do. So it's a little bit ironic that now I'm studying the stuff I threw away in grad school. So these regions are understudied because they're repetitive, they're difficult to study, but also because they're often considered not very useful or just junk DNA. However, they're really junk, right? So in the 1950s, when Barbara McClintock discovered a transposable element, she called them controlling element because they alter gene expression. 20 years later, Eric Davidson and Roy Brayton, they proposed a theory based on the repetitive nature of these sequences. This is the so-called gene battery model, where they hypothesize these repetitive sequences actually help coordinate gene regulation and help evolve gene regulatory network. because these transposable elements can jump around and make copies of themselves, right? So that actually presents a pretty attractive model to reuse and distribute regulatory logic, therefore accelerating this evolution of gene regulation. So why transposable elements are relevant and important, right? So I'm actually going to bring out this perhaps our favorite example, right? So this agouti mouse has already been featured today in the previous talks, right? This very well-known example of epigenetic impact of diet is actually mediated by a transposable element, right? So we all love these two animals, right? You know, they're so lovely, you know, you almost want to, you know, snuggle with them. But the important thing is, you know, these two animals, they look very different, right? So the one on the left, he's chubbier, his golden fur, but this animal also have a lot of health issues, including high risk of cancer, obesity, and reduced lifespan. The animal on the right is, you know, by every measurement wild type. These two animals are genetically identical, right? So the only difference between them, in fact, is their mother's diet, right? So when their mothers were pregnant, the animal on the left, you know, the mother only ate the normal mouse chow, whereas the animal on the right had vitamin supplement, including folic acid and B12. So this is a perfect example to say, you know, you're not what you are, you're what your mother eats, right? So give us a lot of really good excuses to blame our parents for the things that we can't achieve in our life. My daughter learned this from the lectures I gave, so they use they use that against me all the time now. So what's the molecular mechanism behind this? We all know this is caused by a agouti gene, but the causal variant is actually a transposable element insertion, right? So this IAP element inserted about 100 kb upstream of a agouti gene. This is a transposable element. It can serve as an alternative or cryptic promoter that drive agouti abnormal expression. When the mother takes folic acid as a vitamin supplement, this is actually a messyl donor. So the baby is able to methylate the AP element. And then this element cannot serve as a cryptic promoter anymore. Agouti gene expression returns to normal. You ended up getting a good phenotype, right? So this is a classic epigenetic example. You know, as a function of environmental impact, in this case, mother's diet. But what intrigued me and, you know, students in my lab is this happens on a transposable element, whereas half of our genome and also half of mouse genome are made of these transposable elements, right? So one can only imagine how many diseases would have very similar mechanism, being that, you know, it's not a genetic cause, it's an epigenetic cause, right? So this abnormal or gene expression, which is causal for disease, is not due to a genetic factor, but rather due to an epigenetic factor, right? So keeping this in mind, we actually launched a study now, you know, 7-8 years ago to hunt for the human agouti gene, right? So we call this the oncogene agouti paradigm. So we there are a few we published a few papers to describe our findings. So we actually started to look into cancer, right? The reason is, you know, there in cancer genomics, there's a lot of publicly available multi-omics data. So we decided to start from there. We know cancer is a genetic disease and is also an epigenetic disease. Cancer evolves both genetically and epigenetically, right? And the environment has direct impact on both the genome and the epigenome. Now, one thing cancer cell has to do is to upregulate their oncogenes, right? So we've known this and investigated the oncogene overexpression for decades. Classic mechanisms are written in textbooks, right? So mutations, amplifications, and translocations. These are classic mechanisms to upregulate oncogenes. What we decided to look for is this epigenetic mutation happening in transposable element. I would believe this is actually a major mechanism to activate oncogene. Typically, transposable elements are methylated and repressed in the somatic genome. But during this tumor genesis, right? So there's this epigenetic insult. So this not only mutated the genome, it also mutated the epigenome, right? So one of the very common consequence of this is loss of repression or loss of methylation over transposable element, right? So when these transposable elements lose DNA methylation or other repressive epigenetic marks, they start to transcribe themselves. Sometimes the transcription goes into downstream nearby genes. This is precisely how agouti gene was activated, and we hypothesized that this can activate nearby oncogenes and result in oncogene overexpression. So this is actually known. So there are examples reported in the literature, but we're interested in understanding the commonality. How common is this particular epigenetic mechanism? And the result is very stunning, right? So in this case, we integrated a large amount of transcriptomic data and epigenomic data from TCGA, the Cancer Genome Atlas project, as well as from the Roadmap project. And we found that at least 50% of tumors that we looked at had at least one such event that we later call oncoexaptation. So this is defined by a highly expressed oncogene, but there's really no mutations or amplifications or translocations around this oncogene. If you look at where the transcription starts, it actually does not start from its own promoter. Rather, it starts from a nearby transposable element that lost DNA methylation, right? So basically, these oncogenes are highly expressed because these oncogenes actually picked up a new promoter, a novel promoter to drive it, and transposable element provide such promoters. This is pretty widely spread, as I mentioned, 50% of the tumor we looked at had at least one oncogene that is expressed this way. And this event is recurrent across patients, even recurrent across tumor types. These are some of the examples. I'm just going to use the last example here, number 10 on the list on the bottom here as a quick illustration. So this one we call AlloJB Lin20B, right? So Lin20B is a bona fide oncogene represented by this blue exons. It is actually highly expressed in a lot of cancers, especially liver cancer. But we found that these highly expressed Lin20B, the transcription does not start from its own promoter. Rather, it starts from an upstream AlloJB element, right? So this AlloJB element loses DNA methylation, start to transcribe, and go into the downstream Lin20B gene, resulting in its overexpression. So this is just like the agouti gene example. So what's the implication of this, right? Turns out this event already by itself already have prognostic value. Again, let's just focus on the first panel here, AlloJB Lin20B, right? If we separate patients based on the type of Lin20B they express, blue represents canonical and red represents this AlloJB version of Lin20B, right? So in liver cancer, patients with this AlloJB Lin20B expression features much worse. This is not difficult to explain because these patients are expressing a much higher level in oncogene, right? So going back and look at the epigenome, right? So we look at the canonical promoter of Lin20B and this AlloJB transposable element, and we use multi-omics genomic technology. So here using KH-seq, this is a type of RNA-seq that capture 5' end of the transcript. We can see that the transcription indeed start from the AlloJB element. If we zoom in and look at this particular AlloJB element highlighted by this red box here, we see classic promoter epigenomic signature. If you look at K27 acetylation, K4 monomethylation, K4 trimethylation, K9 acetylation, or attack sequence, you see this classic promoter activity happening over this transposable element. Whereas if you look at, so this is happening in cancer cells, right? But if you look at the normal cells, this is the data collected by the Roadmap Epigenome Project, demethylation pattern across different tissue types. And you'll see in all the somatic cells, and this region is methylated, almost like, boring, right? So they're heavily methylated, right? So further, we want to make sure that this is indeed causal. So in this case, we used CRISPR. Now technologies, come along. CRISPR technologies come along. So in this case, everybody uses CRISPR to take out, important genes or regulatory elements. A few groups, our group included, use CRISPR to take out transposed element or junk DNA, right? So this experiment is we're using xenograph as a model to look at the potential of these cells to form tumor. Wild type cells form tumor readily. If we delete Lin20B promoter, right, these cells still able to form tumor, right? It's just a little bit smaller, but they can still form tumor. However, if we delete this AlloJB element, this transposable element or junk DNA, we see the cells actually lose its ability to produce tumor, right? So this argues that this AlloJB is the genetic determinants of this tumorigenic capacity. What about epigenetics, right? So again, genomic technology has really been booming, and epigenetic editing is a possibility now, even for regions like transposable elements that are very difficult to target. Here I'm illustrating just two experiments. On the left side, we're trying to use CRISPR, that CRISPR to bring in a DNA methyltransferase 3A to specifically methylate this AlloJB. So it turns out this is a pretty difficult experiment. We tried it many times, and all we can do is to increase methylation by about 30% and reduce gene expression of Lin20B by about 40%, right? This is also easy to understand because, Lin20B is pro-growth, so you're selecting against yourself, right, in this particular CRISPR experiment. But as a proof of principle, we show that we can manipulate an epigenome and bring down some some Lin20B expression. This already hints potential therapeutic strategies, right? On the other hand, we take a cell line that has methylated AlloJB, and we bring in a site-specific demethylation event, and we were able to, demethylate it at this AlloJB and massively induce the expression of Lin20B. So this experiment actually illustrates how transposable elements can drive tumor evolution epigenetically, right? So it's not like in that experiment, the CRISPR efficacy, CRISPR efficiency is higher than the other experiment. It's really because these demethylated epi alleles drive higher Lin20B expression, and that resulted in clonal expression, right? But this is a epiclone, right? So this experiment actually illustrates that transposable element might be driving this epigenetic clonal expansion in tumorigenesis. So this is a kind of a brief description of the phenomenon, and we get into a little bit to the mechanism. But what about translational applications, right? So we're very interested in the basic science, but we also care a lot about patients and their health. So because this event that I just described is generate a chimeric transcript coming from transposon and going to a downstream gene, right? This is a cancer-specific event. Therefore, it provides a unique opportunity for us to target, right? So one approach that my students are trying is to use a antisense oligo to specifically target this transposon-derived fraction in order to bring down Lin20B expression. And in a preclinical model, we show that this works like a charm. Very briefly on the left, this is a growth assay. Typically, the tumor cells will grow. You can kill tumor cells with pyrimycin, that's a positive control. If you use scrambled oligos, they don't kill cancer cells. But if you use antisense oligos that target those LGB elements, you can actually stop cancer cells from growing. And this event is specific to this version of Lin20B, right? So if the cells do not have this LGB Lin20B, they still grow, right? So this offers a kind of a very novel alternative way of targeting cancer cells. So this is pretty cool, right? But this is actually not the coolest part, right? So it's the same event that we're looking at. Transposon, get demethylated and express themselves, drive overexpression of chimeric transcript in cancer. Now, these chimeric transcripts sometimes translate into proteins and present chimeric proteins, right? What are these chimeric proteins? They can be used as new antigens. So all of a sudden, we're turning this around and then see whether or not transposable elements are providing novel antigens that potentially allow us to target. So this time, we're again integrating data generated by the Cancer Genome Atlas, GTAC, as well as Phantom. And we recognize that almost every single tumor cases that we look at 10,000 of them, they have at least one such candidate, meaning that they have this tumor-specific chimeric transcript coming from transposon going into the gene, and they potentially generate a new isotope that we can target, right? So, the next round of data integration reach out to the mass spec community. So here, I'm going to try to speed up a little bit. I think I'm running low on time. So we show that these are not just our imaginations. These chimeric transcripts, they indeed encode protein because we can find them from mass spec data. Not only that, we can also find them by doing MHC pull down followed by mass spec. We can identify those specific new peptide, right? So this is extremely exciting. The reason is, as I mentioned, this is an epigenetic event, right? So they are actually much more recurrent across patients or even across different cancer types. Unlike mutation-derived new antigen, they're very, very patient-specific. So we're motivated to think about, creating a vaccine cocktail that may be able to target many different cancer types. So this is kind of some of the ongoing work in the lab, right? So this is very cool. It's still not the coolest thing, right? So the real cool thing is, again, epigenetics is reversible, epigenome is targetable. So this presents an opportunity for us to, what if there's no new antigen, right? So maybe we can come up with a way to specifically target an epigenome to trick cancer cells, but not the normal cells, to produce new antigens kind of on target, right? So this is also some very exciting areas that we're exploring. Recently, we published a review article at Nature Reviews Cancer to describe this novel ways of targeting transposable elements for cancer therapy, all started from agouti mouse, right? I encourage you to read this paper. If you read this paper, you're my best friend. All right, so I'm gonna, bring you out of this case study and kind of just show you a little bit about my own experience with all this consortium work, right? I started as a student, learning the Human Genome Project. In fact, when Roadmap started, I was a postdoc. So when I started work with Fred, I was a postdoc working on the Roadmap project. And later on, I was fortunate enough to be involved in ENCODE and Target. And then I want to now just finish up by very briefly introducing a few ongoing multi-omics consortium projects that I feel is very relevant to our discussion. Okay. So first, obviously, I want to mention our own target project, right? So NAEHS launched the target project, and we're fortunate to be part of target 2. So target 2 tries to define a paradigm where we can systematically investigate the epigenetic impact of environmental exposure, right? So the goals of target 2 are twofold, at least twofold. First, given a specific exposure, how does the epigenome respond in target tissues? And second, can we learn the target epigenome change by studying a surrogate tissue? So the consortium decided to use mouse model to implement several exposure paradigms and to look at several toxicant, including arsenic, BPA, and also PM 2.5, among others. We have generated thousands of epigenomic data sets, and the consortium right now is putting together the final data release and multiple manuscripts as a package. So this year, I think it's gonna be a big year for Target. So this is really just a quick shout out to this NAEHS initiated project. I want to quickly bring us back to the human genome, right? So the human genome obviously is very, very impactful, but that human genome reference, that book that we all use, it has limitations, right? So we talked about it being not complete. There are still sequences missing, but more importantly, across its entire span, the reference genome only represents the sequence of just one human haplotype, and not a real haplotype. It's actually a mosaic haplotype. Oftentimes people ask, the human genome reference we use, whose genome is it, right? So it's actually a mixture of multiple individuals that were recruited at the beginning of the Human Genome Project. Therefore, this single monoploid reference structure is actually now a critical barrier to representing the broad genetic diversity in the human population. A little bit over 4 years ago, this Human Pangenome Reference Consortium was launched to produce a complete reference of human genome diversity, right? So the current goal is to produce 350 genomes, haplotype resolved. So that's going to be 700 haplotypes. Hopefully, most of them are telomere to telomere, and this will allow us to incorporate human genetic diversity into the reference. So this human reference book, right, it's going to grow, and we're going to provide this pangenome reference for upcoming biomedical sciences. So there are a series of publications that came out over the past couple of years to illustrate the progress that we have made. So the Pangenome Project, at the end of the day, is a collection of germline variations. We actually have become really good with identifying genetic variations among individuals. However, we're still pretty bad at understanding the functional consequence of these variations. So a couple of years ago, a new consortium called Impact of Genetic Variation on Function was initiated. And this is an NHGRI project. And this project has already started engaging with many other consortiums to focus on understanding the genetic variation on function, right? So we're developing all sorts of paradigms to systematically evaluate genetic variation and how they, respond differently to in different contexts, right? So this is a kind of a relatively new 2.5 year project. Moving along, last year, not even a year ago, a common fund project started. This is called somatic mosaicism across human tissues. Right. So we've been talking about germline variation. We've been talking about the human reference genome, but we also know in our cells, the cells actually don't share the same genome. There are somatic mutations, and everybody is actually a somatic mosaic, right? In the context of transposon jumping, transposon can jump somatically and form this mosaic body that everybody actually is, right? So this project started last year. The goal is to resolve a conflict in the field, right? So we all agree that there are somatic variations, but people actually have a hard time agree with each other. What is the frequency, right? What is the tissue specificity? What's the dependency on age, right? So the hope is this network will generate a map, right? So yet another map, another resource that describe the mosaicism across tissues and across multiple individuals, right? So again, this project is still at the planning age. We're doing benchmarking. We're not even a year old, but this is up and coming. I'm going to use the last project as my summary. Okay, so this is a very recent project, only a few months old. And this is a collaboration at this point between NCI, NHGRI, and of course, NAEHS, right? Because this is really the theme of my presentation here. The consortium is called Multi-Omics for Health and Disease. So this consortium aims to advance the application of multi-omics technology to study health and disease in ancestrally diverse populations, right? So multi-omics analysis can help define relationship between different types of omic data and unravel biological networks regulating transition from health to disease. So this specific initiative is expected to produce consensus approaches, best practices, and standards that can be generalized across different diseases and populations. It will also generate a standardized and harmonized data sets for general research use, right? So that's going to be available through control access processes, as well as through a portal for data visualization. And hopefully our virtual epigenome browser will play a role in that mission. And ultimately, I think this project, this whole program, will enhance the utility of omics technologies in understanding the biology of health and disease. And that's the ultimate goal of integrating multi-omic data to understand our genome. With that, I would like to just quickly acknowledge my lab. These are a group of young, energetic, and brave investigators and never rest. I work quite a bit in the context of consortia. I started as a postdoc in Roadmap Epigenomic project, and now I'm very heavily involved in several of the consortium projects that I briefly described. With that, I'd like to thank you again for your attention, and thank you again for the opportunity to share our research here. Thank you very much.
Thank you, Ting. It was really quite a tour de force. Really very exciting stuff.
Thank you.
So we have the opportunity now to engage in a discussion that will allow us to really focus on this question about modifications of nucleic acid, particularly focusing on the RNA rather than DNA. But of course, as Ting and others have shown, these things are intimately related. And so, Rick, I don't know if you have any comments you want to kick us off with. I know at least one of these models is near and dear to your heart. And then we can engage with the council as you wish.
No, thanks, Trevor and Ting. Great job. It's as you know, good old agouti is a mouse model that we've been studying for quite some time. So that's it's really quite good to see how these fusion transcripts with repetitive elements are really beginning to take shape. One question that I have, and this goes back to your multi-omics approach. It's really so how concerned are you with the heterogeneity of the data sets, especially for proteomics, for metabolomics, so you may have a proteomics data set, but depending upon the technology that you use to actually study it, you may there may be a great deal of functional heterogeneity. So how big of a problem is that for this whole field?
Thank you for a great question. It is absolutely a big problem, right? But just as any problem is also an opportunity, right? So I think there are everybody is aware of this type of problem, and we're coming up with different type of approaches, new technologies to begin to address this. You've heard of single cell, right? So single cell is taking over the field. Single cell technology is not perfect yet, but really the number one thing single cell technology allows you to address is heterogeneity, right? So I certainly have great hope in sort of a single cell breakthroughs to help solve these problems. Even before that, we're creating reference panels, right? So sometimes reference panels allows us to create models. So even though the data has heterogeneity in it, we can kind of deconvolute the data and then be able to dissect the relationship, the nuances contributed by different cell types. So I would say you're absolutely right. This is a very big problem, but we're tackling it, and every problem is an opportunity.
Great, thank you, Ting. Just a quick reminder, I think we have all of our speakers available, so we're able to cover the full gamut of topics addressed during the symposium. And if we use the raise hand as Karen has done, I'll be able to follow and track as we move along. So Karen, if you want to kick us off, please.
Yeah, thank you. Ting, I wanted to ask you a question, and then I had a comment, and it goes along the lines with what Rick just mentioned. One thing, for example, you mentioned that you'd be looking at different tissue types, or you look at different samples, and we also we study repetitive elements, not for just their regulation and transcription, but for their formation of alternative DNA structures, right? And these are not just in lines and signs, but everywhere, very abundant in the genome. So my comment is, so we study, for example, in mice, we looked at tissue-specific effects of aging on genetic instability at these non-B regions of endogenous instability or of obesity, things like that. And we always get comments from reviewers on grant proposals, on manuscripts. Well, what about, so we say we see a big difference in spleen with HDNA versus BDNA, right? There's much more genetic instability in spleen. And then they'll say, okay, but the spleen is made up of so many different cell types. So what about individual cell types? And you sort of address that about single cell analyses, but I really think at some point to understand, very mechanistically. So I'm a biochemist trying to do biochemistry in a mouse. And so we do it at the tissue extract level, but I think at some point it would require individual cell types, if not individual cells, though that is cost prohibitive for most studies, for biological studies. But what do you think about that, about the different cell types, and how when you look at a spleen sample, it's representative of so many different things that you may be like losing effects because they kind of average out?
Yes, you're absolutely right. This is a key question. I think people have been investigating, struggling, if you will, for many, many years. In fact, when I was in school, right, when I started learning epigenetics, they always say your epigenetics is only as good as how pure your samples are.
Right.
Right? So in what, I think one of the goals of creating, if you will, reference panels, right? So, if you have the ability to purify specific cell types and create that kind of reference panels, that can be useful. I think that's the motivation behind the old roadmap genomics project before, single cells are kind of are dominant, right? The other thing is, I think, the same DNA definitely behave differently in different environment, right? So this trans environment is not just, the trans environment has many, many other factors, right? So the heterogeneity is also defined in, there's just a lot of nuances in how to define this.
No, it's very complex. Very good. But let me
Right.
throw one more thing in there. So you were talking about the lines and signs and sort of these transposable elements, which are really important in many aspects, right, that we used to call junk DNA. We know that that's not absolute at all. But you mentioned mostly in this context of transcription and oncogene expression, tumor suppressor, tumor suppressors being, inhibited, et cetera, in cancer. But as I mentioned, we study non-BDNA and alternative DNA structures at these repetitive elements.
Yeah.
And they also lead to things like, well, they regulate replication, they can regulate recombination, and they certainly, in our area, is they can stimulate genetic instability, and largely in the form of double-strand breaks and deletions. And so that could also contribute, I guess, of course, to transcription. But is it more than just the transcription and regulation that you're interested in? Because clearly cancer also involves mutations and genetic instability in how they alter transcription, but other things too, like losing important regulators and elements that go beyond transcription. Are you looking at that? Because you have the perfect data sets and venues to do that. And if you're not doing it, we'll collaborate with you and do it.
I love collaboration. So, I mean, if you think about this, right? So we always look at the problem from different angles, right? So on one hand, we're trying to be a reductionist, try to isolate a specific problem to work on. On the other hand, these are clearly systems levels biology, right? So it does require, perfectly fitting into like the topic that I attempt to present, integration. Right, so put them together, right? So I absolutely see the opportunity to collaborate. But the work that I presented is somewhat, reductionist. We're going to focus on that particular aspect, so our work was actually powered a whole lot by thinking about evolution, thinking about how epigenome has been evolving, how transposon have been evolving epigenome during the course of evolution. And then cancer is just a, accelerated evolution, right? So once you have that concept incorporated, it becomes relatively easy tractable. It's still not fully tractable, but it becomes tractable. So that's where we gain some traction. I think the integration part is exciting, but of course the challenge is, can you truly track every parameter, every kind of variable in this in the
with
context.
your data sets, then it's easier to bring it, like in these large consortia, and you have a data set so that we can all apply our areas to that go beyond regulating transcription, for example. But I think that's an area that we should consider, that has been considered, but should be more carefully considered, because it is critical, like you said. It's a whole systems approach. But thank you for the great talk. Excellent.
Sure. Thank you. Thank you.
Trevor Penning.
Oh, hi. Thank you, Trevor. I had to say that. I had two general comments, and it's actually for all of the speakers, and they can all jump in. So first of all, let me say what a tour de force today, just quite remarkable. We've heard, so my two comments are as follows. We've heard a lot about methylation of DNA, methylation of RNA, methyltransferases. And at the end of the day, all of this is dependent upon a methyl donor, which is SAM, of course. And SAM gets its methyl group from folic acid. And so I'd like to hear people's opinions about how important it is for us to be studying folic acid. We had the example of the agouti mouse earlier, but maybe this is a general phenomena that we have to start thinking about, of the interplay between diet and these downstream effects. So that's one comment. Why people think about that comment, maybe I'll make my second one, if I'm allowed.
I think Tom maybe has an answer or a response for you before.
Yeah, I think that's a great question, Trevor. And I think, we're seeing in our studies, not with folic acid, but with cueing, dietary factors that influence sort of exposure responses, I think are going to be immensely important. I would say, yeah, I think, folic acid is going to be a key player in a lot of what we think about on the modification world. And that definitely has to be factored into our studies. I don't know how to do it, but I do think it's a good idea.
Your second point, Trevor.
Okay, my second point is I know that we were focusing on RNA today, but I'd like to make a comment about DNA, if I may. you know, we often think about DNA and we think perhaps a little bit simplistically about CPG islands and how the cytosine might actually be methylated. And of course, we also all know there's more than just 5-methylcytosine. There's hydroxymethyl and the formyl cytosine as well. So we have a number of players on the C residue, but we mustn't forget about the G residue. The G residue can go oxidative modification. And we always think about 8-oxoguanine, but 8-oxoguanine is an intermediate. It can go on to other things like fatty allox. And so I think there is a DNA epigenetic code when you start thinking about the combinatorial capacity that we have to think about as well.
Any takers? Tom.
I'll defer to the group, but if I'm happy to jump in and say, you know, Trevor, I totally agree with you. I think, you know, I've read some of the adaptomic studies, and the diversity of lesions and epigenetic marks that are found on DNA is something that we have to you know, think about. It is combinatorial. It is super complex. You know, it sort of brings up the question of what should we really be training our students to do, and you know, big data analytics, I think, is something that we really got to do more of. So you know, I'll end there.
Maybe I want to add a little bit. Yeah, that's a very interesting point, Trevor. But you know, one key difference between the C modification you're referring to and the G oxidation you're talking about is that, you know, for C modification, there are dedicated enzymes like TET family enzyme that can catalyze it, right? But for G oxidation, like fatty acids, G, you know, that's mainly generated by ROS, not catalyzed by enzymes.
Right.
So in other, there could be selectivity issue, right? You know, one is pleiotropic, the other is maybe direct to certain, like a CPG island and so on. Yeah. So there's one difference there.
Yeah, what I was getting at was that, you know, we focus on the methylation of cytosine, but what happens if that cytosine is being methylated right next to an 8-oxoguanine residue, right? You have a change, you have a double change in the code at that point, right? Well, not the DNA code, but the DNA modification. You have two modifications right next to each other, and when you can think about how many ways cytosine can be modified and how many ways guanine can be modified, then you have a new code to think about, right?
Yeah, true, but the frequency is very different. You know, like a 5-HMC, you're talking 100 ppm, but G and fatty, and you're talking about 1 ppm, a sub ppm in most cases. Yeah.
Point taken.
Yeah, good. Dr. Mutlu.
Thank you. Thank you, Trevor. First, I would like to thank all the speakers for their great presentation. I learned a lot about epigenetics and RNA epigenetics specifically. I have two questions. One for Tom. Hi, Tom. It's been great seeing you after so many years. So you mentioned the Q-base being dependent on the microbiome for humans, right? You know, so are there any studies that looked at, let's say, immune responses in patients who receive antibiotics, whether you replace the Q-base or not, you know, in the diet? Does it make any difference?
That's a great question, and great to see you again. I'm not aware of any studies with regard to that, but you know, I think it's a great question to ask, and you know, and to detail. There are some Q salvage pathways that can be used to, you know, to bring it back into play in the cell, but you know, I think whenever there's an antibiotic in use and you're changing your gut microbiome, you could have the potential to change not only Q-base, but I mean, but other things that could influence the epitranscriptome. I'll also note that, you know, from a diet perspective, I think dietary sources of the Q-base, you know, are variable. It's like in sour cream and things like that. And There's a group in Ireland that sort of looked at Q levels based on diet, and it dramatically changes. So.
Thank you. The other question is for Justin, as well as anyone who does single cell RNA sequencing. One of the issues that we encounter when we do single cell RNA sequencing is the sample preparation. So I'm not sure it's standardized even for each organ that are being studied. So do you know if there are any efforts to do that? Because I think just the preparation itself will introduce a lot of variability, at least in our class.
Yeah, Gokhan, great question. Nice to see you. So this is a big challenge. I didn't have a chance to go into too much detail, but the phenomenon that Gokhan is referring to, kind of like even within a individual prep, is that the choices you make in your dissociation, sort of like the physical dissociation, doing the enzymes you're including in your mixture, how long you're dissociating, can have very substantial results on the data you're getting back. So we experienced this firsthand with one of our first studies, actually based out of Target, that Ting was talking about, where we had taken some of the hippocampuses from our lead exposed mice in Target, from some of our extra samples, and we dissociated them. We ran single cell and almost got no neurons, right? Because of that was the method that we had used for the dissociation. So there are ways that we can sort of get around this. One way, Gokhan, that folks are thinking about it is with like single nuclei, where we can take a frozen tissue, isolate the nuclei. We're not going to see that same bias necessarily that we might see. And then there's other new methods that are coming down the pipe. I don't know if folks have had a chance to try. So 10x Genomics has a new kit they call the Flex method. And we've used this where you take a frozen tissue, then you fix it, then dissociate. And we've gotten pretty good representation of the different cell types in that method. But I think you're totally right. And I think it speaks back to what Ting was talking about, and even like Karen's question about like the heterogeneity issue, that this is something we have to have a good handle on when we're interpreting these various data sets, especially as we're thinking about like, do toxicants push cell type proportions in different directions?
Thank you, Justin.
Thanks. Yeah. So some of us might say that actually reflects what we call biochemistry in terms of generating samples. Karen.
Yeah, I just wanted to go back, revisit Trevor's comment. So we think about, so he mentioned about DNA, of course, and we were talking about RNA, but also we need to think about nucleotide pools, right? So sometimes, I don't know, I tend to forget about those. I think about DNA mostly and then RNA next. But, all of the pools have to be, they are exposed to these things as well. Toxicants, reactive oxygen species, they're modified, then they're incorporated. They can be incorporated into DNA or misincorporate or not incorporate and inhibit replication, etc. So all of these things, it seems so complex, and it almost seems like, Trevor mentioned looking at folate, for example, in methylation as an overall arching sort of player. But what about looking at enzymes that modify DNA and RNA? Because that way, or looking at those levels, increasing, decreasing, and then seeing how the DNA and RNA both are modified, or the nucleotide pools as well, because that way then we could maybe say, I'm just trying to think of how to like dissect this because it is so complex, but maybe overlaying DNA with RNA modifications and seeing how those are influenced by enzymes. It seems like to me, if you look at all of the different types of modifications that could occur at any given base in 3 billion base pairs, and then all of the RNA modifications, that maybe the enzymes are an easier study, and then seeing how those regulate this overlay. And if one DNA modification alters an RNA outcome at that gene level, I don't know, somehow it seems like we have to figure out a smart way, like all of you who just spoke today could maybe think about, and the computational expertise, artificial intelligence utilization in trying to come up with some way to look at at a common denominator that's less overwhelming. And maybe that's DNA, RNA modifying enzymes. I don't know, just a thought. But I'd like to hear others discuss this, because I don't know where to begin with all the different, there would be millions and millions and millions of potential modifications to DNA and RNA at any given point in time. It's just, it's overwhelming, but critical. I mean, that's what regulates everything, right? So we have to understand it, but how?
Maybe
That's
Yin
my
Yang has
question.
an
How?
answer
That's
for
to the rest of you. How do we do that?
you. Dr. Wang, please.
Yeah, Karen, you raised a very good point. Yeah. So for the LKPG3, we found to be regulators, for example, by repeat RNA. You can not only demethylate RNA nucleoside, it's also DNA repair enzyme. You can repair alkylation damage in DNA. So that's one thing we can think about, right? Not only impact RNA, but it may also impact DNA repair. Another thing is that there are several years ago there are several studies they showed that the RNA M6A actually can be metabolized and then delivered to the nucleotide pool. So then ultimately can be incorporated in the DNA. So some of the N6 methyl A in DNA is thought to be from the metabolism of M6A in RNA. Right? So that's another thing we're thinking about. But that's a very good point. Yeah.
If we can add to the complexity of the conversation, maybe I will invite Victor or Yvonne to speak a little bit about contributions of sort of the protein components of the nucleus, either histones or architecture of regulatory material.
Sorry, I didn't get your question again. Sorry.
No, so I was saying we've talked a lot, of course, during the symposium about the modifications of RNA and DNA and others, but of course that takes place within the context of an architecture that's chromatin. And I wondered, and of course in your presentation, Yvonne, you spoke about histone isoform and that contribution. So I just thought we would expand the conversation to include those aspects of the epigenome, epitranscriptome.
So I mean, that's a very amazing way to look at it, in the fact that we have these variants and we don't know their modifications. We've started also analyzing them, and we're seeing that the differential modifications as cells go through this arsenic-mediated carcinogenesis. And so it's still like a brand new area, trying to understand how those modifications themselves could contribute to changes in gene expression. So it's really still a lot of things that we have to find out, and we don't have that information yet. And you could see also that that could change the chromatin architecture, right? So we are, from my biophysical studies, we're seeing that the different variants, just one amino acid change, and it's actually changing how dynamic that nucleosome is. But the question is that where are they located, which also is an issue. We still don't have that answer yet, but hopefully in a year we'll be able to know that.
Great.
I don't have any great insights to answer that question. So, we talk about some of these things. And to me, an important question is, why do we have such a specific phenotypes? Is it because there are changes genome-wide in whatever, DNA methylation, histone modifications, in whatever RNA modifications, in whatever of the different things that we've mentioned today? Are the genome-wide changes, but we only see a few phenotypes because the other ones are not as important? Or are the changes very specific in the specific genes? And so from my perspective, in reference to your question, if there is a sequence specificity where some environmental effect is affecting only a few sites in the genome, it has to be through a mechanism that recognizes DNA sequence, and that is DNA binding proteins, transcription factors. You were addressing the issue of chromatin organization, Trevor. That was your specific question. I think CTCF is which is the main protein involved in this. It is poised. We keep finding it continuously. It doesn't matter what we expose to different things, and CTCF at different sites in the genome is always in there. And I think it may be a very sensitive protein to play this role because it's exquisitely sensitive to DNA methylation, has all these fingers like Yvonne mentioned, and they have different roles. So that depending on which one is affected, then it's affecting only a subset of sites in the genome. I think some of the fingers are involved in the residence time rather than sequence specificity. And this may also have in regulating residence time on the DNA. And that means stability of loops, and that means frequency of enhancer promoter interactions. So my bias is that transcription factors in general, proteins that bind DNA are very important to consider before we consider general effects on, for example, all DNMTs. You know, I think this idea came up, I think it was Karen that would mention it. You know, DNMTs, there's a ton of them, of protein on the chromatin. And if you mess up the enzymes, I think you will have a very generalized effect through the whole genome with very pleiotropic phenotypes and very, you know, it may be lethal in those cases, it may cause lethality, and we just don't see a specific phenotype and we are not paying attention. I don't know, I'm just rambling.
No, very interesting thoughts. Are there comments or questions? Bless you. Karen, please.
I have a question for Yvonne. I thought your talk was really fascinating, and I don't know that much about histones and the histone variants, but I know a lot about DNA structure. So when you talked about the histone, the variant that altered the size, if you will, the nucleosome, does that also alter the size of the linker region? And if so, did you notice that there are more double strand breaks, or did you do like nuclease probing? Is there more hypersensitivity to reagents, to nucleases within those altered nucleosome regions?
So we've not done that because we've had issues with, like I said, we just have the CRISPR-Cas9 cell lines now tagged because we need to know where they exactly go to. We tried doing ATAC-seq, but we just saw ATAC-seq everywhere. I mean, so we don't know whether it's specific to those nucleosomes that were variant tagged or not. So we're still doing that experiment to test. So all what I showed you now was mainly with in vitro system and nothing in vivo yet. And so we don't, we tried doing a simulation with the nucleosomes, and we kind of see that it's kind of it's a very subtle effect on the DNA, kind of pushing it off like that. And so it seems as if when you push on one side, it kind of reacts this way for the DNA to fold over. So that's what we're seeing right now, but we don't have in vivo, we don't have a clear idea of what I just told you right now. What has been really fascinating for us is that we see, while we see some of this in lung cancers, we also see different variants of those in breast cancer. And so the question is that how is a single amino acid change on a histone having this major impact in different cells? We don't know.
Right. But I wonder, you know, like with, well, methylated cytosine, let's talk about that. It doesn't wrap around the histones because of the arginines that get in the way. So maybe if it's altering something like that sort of a charge, that it would allow differential wrapping or interaction.
That's what I thought, but they all wrap in vivo. So there's no issue. They wrap really nicely in vivo. It could be that maybe if we have the in vivo system, which we hopefully should have soon, we could see whether they frequently associate with another type of histone variant on H2A or H3. We don't know that yet. And maybe that would then add to the effect, to the additive effect that we might see a major. I remember when I started doing this and someone told me you're not going to see any changes and you shouldn't even bother doing it. But I'm just a little stubborn on that. And I kept pushing on that. And it's just amazing what we're seeing. We just haven't gotten that into in vivo system yet. Hopefully soon.
Yeah, well, thank you for your tenacity because it's quite interesting. Yeah, thank you.
Rick.
Yeah, I just wanted to add that I've thoroughly enjoyed the presentations. I mean, Fred, thank you for your coordination of getting these spectacular set of speakers together for this this symposium. I mean, one question that I have is that, and we'll talk a little bit more about this tomorrow, is this whole concept of the exposome. I mean, this is exploding now. There's lots of interest in studying, getting beyond studying one exposure at a time, and there are tools that people are developing to measure multiple different exposures. But a very important and a critically important element of the exposome and operationalizing the exposome is to characterize biological responses to these multiple environmental exposures. There's been a lot of interest in incorporating epigenetics into exposomics research. So I just wanted to challenge the group. So how, it's clear that transcriptomics is playing a really important role here. So it's not just modification of DNA. So what are the thoughts? I know Gary just put his camera on. I bet that would capture your attention here. But what are the thoughts about capturing all of this epitranscriptomics data into the exposome framework? I don't know, maybe Gary, do you want to just start off?
Yeah, go ahead, Gary, please.
Yeah, I mean, I like to Rick's point there is that, to me, a lot of these changes that were described today, it's a very reliable biological output that can be done on an omic scale. And as we get the sort of exposomic level analysis of the complex exposures, I think about the value of having a functional readout of the exposome, having like a functional exposomics. And it would seem that the epigenome sphere would be a very good place to help us prioritize the exposures that have the largest impact on the epigenome and whatever level of regulation we're talking about. And so it gets to this sort of multi-omics analysis, is like, how can we bring in the exposomic as a layer and look at that as a way to say, identify and prioritize the exposures that have these very substantial and lasting, impacts on the epigenome. So yeah, I'd just be curious to see about how, people have thought about thinking in more than just one or two types of exposures, but really getting a complex exposure and using the epigenome as a way to prioritize that biological response.
Perfect question. Victor, please.
I think that, that's a great idea, but it becomes the issue is what tissues do you look at all the tissues? Do you look at them in a cell type specific manner? It becomes really daunting to do this, and like you expose the organism directly. You have to look at the brain, the heart, the muscles, the bones, the liver, and then you will have to hopefully eventually do it in a cell specific way, so that you can capture. We were talking about this before. If one cell type that has more DNA methylation and in the same tissue, another cell type has less DNA methylation as a consequence of an exposure, it would get confusing. So we have done, and we are doing something like that with the sperm. It's just that one cell type, and it's interesting. You can expose an organism to different chemicals, and then in utero, and then look at changes, and you see epigenetic signatures that are specific for the exposure. So I think, it's a great idea, but it's very complicated.
And I think that, some of the work of the target consortia, like where we can think of like in peripheral measures, might be that starting point when you think about, extracellular vesicles and what can be contained there, is that the exposomics initially is being measured in blood, and what are the sort of peripheral markers that may be out there to help you direct to those organs and cell specific changes or something in there. But I think that's where like getting that connection would be, I think, a very nice link.
So I think I'll turn to Tom for the next question. And after that, I'll ask Fred to close us out for this session of the discussion. So Tom?
Yeah, mine wasn't really a question. It was more of a response to the exposome question. But I love the idea, and I agree with Victor. It's a daunting data analytics and with samples. There's a lot there, but I do like the idea of connecting the exposome to the epigenome, and then using that as a filter to get to the epitranscriptome. So targeting those RNA modification systems whose writers or erasers are epigenetically regulated, I think we'll see a beautiful loop that from a gene regulation and a phenotype response. So that's where I would suggest we go.
I just wanted to also add comment to what Rick said earlier. I think it is daunting. It's a lot of data, right? But also, when we are trying to correlate, the quantitation is really important, right? So let's say, with exposure, let's say RNA methylation goes up or goes down or whatever. But then if we then miss like where exactly that these things are happening, and if that cannot be quite quantitative enough, I think it can be dangerous that we actually miss the key point. Partly what kind of as per what I was discussing earlier, the specificity. So sometimes I think without knowing the parameters that actually would matter for the downstream effects and things like that, I think just getting so much data without putting also enough effort to make sense of them and then have a mechanism. I think it can be also dangerous to jump to conclusions. So I think we should, as a field, be careful. Yeah.
Thank you, Yunsen. Victor, last comment, then we'll go to Fred.
1 second. And I just wanted to make the point that when we measure the epigenetic outcome of the exposome, we should do it in a cell type that stays with us, because I think, we don't really know what we have been exposed to. Chemicals that last for a long time in us, then we can measure them. But there is certainly a possibility that we are exposed to something we don't know. And it makes an epigenetic change in a cell type that then it is renewed and dies and is not with us anymore. T cells stay with us for a long time. So it would be a great target tissue to look at the exposome. And I think if we can say, if you're exposed to this, then you have these epigenetic changes. If you're exposed to some other chemical, you have these other epigenetic changes. So that if we look at the epigenome of T cells, for example, if we had a one-to-one correspondence between exposures and epigenomic outcomes, we could read the epigenome of somebody's T cells and say this person was exposed to this and this and this and this.
The immunologist will love you, Victor. Fred, please.
Okay, thank you, Trevor. What I wanted to do really was make one blanket comment to all of the speakers. Thank you so much for being generous and sharing your time and responding. And with short notice, you responded very quickly. I thank you for the talks, the presentations. Obviously, they were all great. I think everyone knows that, so I don't have to say that. But thank you so much. I did have a couple of questions, and maybe it'll be better if you respond to me offline. But Yvonne, I want to ask you, and you can tell me later. You don't need to tell me now. But is the SAT-V2 circular RNA modified? Tell me later. And Yun-san, two things I wanted to ask you. One would be, what is the role of cations, magnesium, for example, in with regards to stabilizing sequence? And the other thing that I want you to think about is, we've talked about this before. What might I do to entice you to come over to the dark side?
Right. I don't think we like to think of ourselves as the dark side, but with that, I'll turn it over to David.
So let me add my thanks to everyone. This was everything I had hoped it would be. This was a great day of science. I think it's all work that's very relevant to some of the emerging things that we're trying to accomplish at NAEHS. And I think that you've helped us move our mission forward today. So thank you all for your presentations, and especially thank you, Fred, for your leadership in pulling this day together for us. It was a testament to your commitment to the science, the day that we've had today. So we're going to close out the open session for today. We'll reconvene at 4 o'clock in closed session. And then tomorrow morning at 9 a.m. Eastern time, we'll reconvene for open session with the presentations from Rick and myself on the NIEHS and ERT kind of state of the unions, and then two topical presentations on our environmental justice and health disparities activities, and also on our climate change and health program. So we'll have another maybe not as deep a dive into the science, but hopefully another interesting day for you tomorrow as well. For the staff members and council members, you can stay on the Zoom, but we'll take a 10-minute break and reconvene at 4 o'clock for the closed session. Thank you all.